So, good morning, guys. So, this is the second biology tutorial. Then we are just going to be discussing cell. We've done that already, actually. Then, in relation to its environment, cell and its environment. So that's what we are going to be discussing now. And then, it's not something also very, very cumbersome or so much that you may find difficult to understand. It's just very basic. Just pay your attention and then try to note a lot of stuff. So that's that about that. So let's start. So living cells are known to be surrounded by watery environment. In fact, let me tell you this now. Um, the don't forget, you can assume that life starts with a single cell, right? Yes. So it is also assumed that that single cell is kind of a marine cell. It- When I say marine, it's kind of living in a salty environment. The environment is salty. This information I'm giving is going to help you a lot. So, and if the environment is salty, what's the normal salt we have around us? The table salt, that's sodium chloride, right? Yes. That is why outside every cells, you will have more sodium and chlorine compared to the inside. Let's move on. You get my point. So, living cells are known to be surrounded by watery environment, whether it is marine or not. Shall you get my point? So we shall have a watery environment around cells. So this may include fresh. Some cells are inside fresh water, while some are salt water in which the unicellular organisms live. Intercellular fluid that bathe the bodies of the cell of our animals, of course, intercellular between cells. So it's the fluid in between cells kind of bathe, kind of surrounds the cells, individual cells. The nature of state of matter makes diffusion and osmosis possible. Okay, that means the movement of this water into the cell or some particles from that extracellular intercellular cell intercellular fluid into the cell or outside. That's actually kind of possible because of the nature of matter. And what are these things? These two guys, diffusions and osmosis. For those more than that, you are going to see everything in this course. As we all know, matter can be defined as any material that has mass and occupies space. Okay, we've discussed that earlier in chemistry actually. It includes all living and non-living things around us. That's matter. When I was discussing this, I mentioned that living organism is a matter, as in biology. You are seeing it in action now. So matter is also made up of what? Tiny particles, which may be molecules or ions, not ions. That's an autocorrection. So ions and can exist in three forms: the solid, liquid, or gas. Although you know that we also have the plasma and other guys too. Okay, so let's now discuss diffusion. So diffusion is the process by which molecules of substance moves from a region of higher concentration to a region of lower concentration. It can be through a medium of air or liquid until equilibrium is reached. Look at that definition. Diffusion is what is the movement. Let me just say that movement of molecules from a region where they are more to a region where they are lesser. Do you get my point? In the sense that um you spray perfume in maybe in one corner of a room. After some time, the perfume kind of circulates the whole room. The perfume kind of diffuses into the whole room. That's diffusion. So diffusion is the sense that eh, we have what movement of molecules. I'm trying to break this down. We have movement of molecules from what? From a region where it is more to a region where it is what? Less. That's the idea. So the difference in concentration of substances in two regions before diffusion occurs is called concentration or diffusion gradient. Okay, good. You can now say that, oh, the region where that guy is more, we have higher concentration gradient. Where it is lower concentration gradient. Do you get my point? That's the idea. So that means diffusion is what? Movement of what? Molecules from region of higher concentration gradient to region of what? Lower concentration. Let's move on. For example, if KMNO4 potassium tetraoxo manganese 7 is placed in a beaker of water and allowed to stand. The purple color of KMnO4 starts spread outward from the crystals. Eventually, the color spread evenly throughout the water medium. That's basically, for those that are familiar with Yoruba, we call this arrow. If you put KMnO4, the arrow inside water, you don't need to stir it. After some time, it diffuses into the whole medium. You see the whole medium being colored. The KMnO4 kind of colors the whole medium. That's the idea. So diffusion just basically means we are molecules in ions or substances are moving from what region of higher concentration to region of what lower concentration. So diffusion is also observed in the spread of odor of perfume, insecticide which is used to spray a room, and also in the release of gas from the anus. You know what that is? So diffusion, process resulting from random motion of molecules by which there is net flow of matter from region of higher concentration to region of low concentration. Okay, a familiar example is perfume of a flower that quickly permeates and the still air of a room. Okay, note that diffusion involves movement of molecules from where they are more to where they are less. This particular note is important in the comprehension of what the data slides. You knowing that it is the movement of molecules from region where they are more to region where they are less. You're going to get a lot of source from that. So, factors affecting this diffusion: change in temperature. The higher the temperature, the faster the rate of diffusion. That means it's normal. Particles are moving, right? And for particles to move, they need kinetic energy. And temperature is what? The average what? The measure of average kinetic energy of everybody. The higher the temperature, the higher the what? Kinetic energy of those particles. Then the higher the kinetic energy, the faster they move. So the rate of diffusion depends on what temperature, in the sense that temperature increases what? The rate of what? Diffusion. The size of the molecules. Diffusion increases with decreasing size of what? Of molecule. It is just for kind of, okay, let's, I don't want to bring in physics inertia, but let's just look at it this way: the bigger the molecules, the heavier they are, the difficulty they get in moving from one point to another. That's the idea. The state of matter, diffusion in gases is definitely faster, because the particles in gases are already moving. They are moving, they are in their random motion already, so the diffusion is expected to be faster. Difference in concentration, that's the gradient, the concentration gradient. So the greater the difference in concentration of molecules, the faster the rate of diffusion. If you have higher concentration gradient, in the sense that there is more somewhere and there is very very less the other side, you will have diffusion faster in that sense. Diffusion importance to life. So in which cases do we have diffusion? Movement of carbon dioxide through the stomata of leaves during the process of photosynthesis. Movement of oxygen into the leaves during respiration. All these are life examples. Movement of water vapor out of the leaf in the process of transpiration. That's also what? Diffusion. Gaseous exchange in lungs of humans. Transpiration. Um, Diffusion. That's done respiration actually. Then intake of oxygen and nutrients and the removal of waste products by the fetus from its mother through placenta. Diffusion. Region from when it is high to where it is low. That's just it. The movement of digested and soluble food from villi of small intestine to the bloodstream. Diffusion. If these materials are more, where they are going to, they won't even go there. That's the concept behind diffusion. Let's move on. The broad division of diffusion. You can see diffusion as the major means of transport of all these guys. That's not divided into what? Imbibition and osmosis. Imbibition is not so widely accepted in biology, but it is important in some biology concepts, like what? Germination. So imbibition is the absorption of one substance by another. In particular, of course, we are absorbing something from, like something is absorbing something. It is because the concentration of that it is absorbing is low in it. That's why it's absorbing it. That's why we also refer to it as what? Diffusion. So in particular, the uptake of water by a plant or seed. When you are planting seed, the seed is going to what? Absorb the water. That's imbibition. You can see imbibition in this concept. You just dip the tip of a newspaper into water, not the whole part of the newspaper, and remove it. You will notice that water will keep moving up until kind of, if there's enough water, it will move up. It will even move to the part where kind of the part where that was not even inserted into the water. That's imbibition. Uptake of what? Uptake of one substance by another. That's the idea. Then the second division is osmosis. You may be like, is osmosis a division of diffusion? Is osmosis not from lower concentration to higher concentration? Let me explain it now. Osmosis is a legit division of diffusion. Osmosis is also the diffusion of water. That's just it. Osmosis is equal to the diffusion of water. And if you have to look at it deeper, don't forget, if you have to look at concentration of something, if I put small salt in a very large volume of water, you see you have low concentration, right? And then if the water is lesser and the salt is even the same quantity, you say you have higher concentration. That means the higher the volume of water, the lower the concentration. That's the idea. So that means when you have the concentration of the solid to be high, the volume of water is low. Or you can say concentration of water is low. But when the concentration of water is high, the concentration of the solute is low. That's the idea. So osmosis, which is diffusion of water, is the movement of water from where water is more, higher concentration of the water, to where water is low. And don't forget, where water is more, there is low concentration of solute there. Where water is low now, there is high concentration of solute. So movement of water from a region where water is low to a region where water is not high. But in osmosis, it is passing through what? A semi-permeable membrane. In some cases, you use what? Selectively permeable membrane. That's the idea. But the main thing you need to get is that the membrane is not allowing the passage like that. There is a what? There is, so sorry for that. There is a barrier. That's the concept. There is a barrier. So moving on, osmosis. Osmosis can be defined as the movement of water molecules from region of lower concentration of solute. This lower concentration is referring to what? The concentration of solute. When they take concentration generally, they are always referring to what? Solute. Do you get my point? To the region of higher concentration of what? Solute too, through a semi-permeable membrane. Take note of the word phrase water molecules, movement of water molecules, diffusion of water molecules. So a permeable membrane allows molecules to pass through it freely, of course, while a selective permeable only allows certain molecules to pass through it. Osmosis will occur only when a semi-permeable membrane separates a weak and strong solution. A weak solution is like a dilute solution. A strong is a most concentrated one. We also need to see osmosis as a type of diffusion where the molecules moving is water. Shey you get? Don't forget in diffusion, the molecules moving are the ions. Do you get my point? So osmosis can then be defined as diffusion of water from region of higher concentration of water to region of lower concentration of water until we attain what? Equilibrium. In osmosis, the diffusion occurs via what? A semi-permeable membrane. So let's look at what permeability is, although I've explained it earlier. So we are just having kind of non-permeable, semi-permeable, and selectively permeable. I've discussed this earlier. You can just pause the video there, read through it. Then tonicity too. When they say tonicity, it is also referring to what? Osmosis. Because you are having a barrier. Then that barrier is going to be separating two what? Two solution. Shey you get? So you can now define this solution based on their what? Relative to each other. Let me just put it that way. So you can have hypertonicity. You mention, okay, let's say we have a cell now in a solution. Don't forget, the inside the cell you are going to have what? Some fluid. Water will be there. Outside the cell there will be some fluid. Then the cell membrane will now be the what? The barrier, right? Which is selectively permeable. So when the fluid surrounding the cell is more concentrated than the inside of the cell, the surrounding fluid is said to be what? Hypertonic to the content of the cell. Hypertonic in the sense that it's kind of um hypertonic in the sense that the concentration of what is surrounding the cell is greater than that which is inside the cell. That's just the idea. So imagine you are putting a cell inside a salt solution. That means you can say that salt solution is hypertonic. But if the concentration inside the cell is more salty, you can now refer to it as what? Hypotonic. Shey you get? That's the next one. Hypotonicity. Hypotonicity is the reverse. where the surrounding fluid, the surrounding solution is what? Of lower concentration. And that's the idea there. So we talk about hypertonicity there. Then the last one, isotonicity, when the concentration of the two guys are the one inside the cell and then the one outside is what? They are equal. That's that about that. There won't be any flow of water. When it is hypertonic, molecules will move from where it is, therefore, of course, remember, movement of water from region of what? Higher concentration of water, where water is more to where water is low. And if water is more somewhere, it is having lower concentration of solutes. So for an hypertonic solution now, the water will move from what? Inside the cell to outside the cell, because the outside the cell is what? More concentrated in solutes. Do you get my point? That's the idea there. It has lower water. That means water will have to go there. And then if water is leaving the cells will tend to shrink. So that therefore hypotonicity, you are placing a cell that is having concentrated inner content inside the solution that is having lower concentration. So that means you have lesser water inside, more water outside. Water will flow inside the cell, then the cell will kind of expand and may even burst. Then isotonicity, there is no flow at all because they are both equal. So importance of osmosis. It takes absorption of water from the soil into the vacuole of the root. All these are they are life examples of what? Osmosis. We can just try and be careful and take a deeper look into it. Then effect of osmosis on biological cells. We have plasmolysis, flaccidity, hemolysis, and turgidity. Let's start. What's plasmolysis? Of course, I've discussed this before. Plasmolysis, you are placing the cell in an hypertonic solution. Hypertonic solution in the sense that water will be leaving the cell going outside. So for plasmolysis now, water is leaving the cell tends to shrink. So that shrinking of a cell that cannot lead to what? The cell death is called what? Plasmolysis. So cells are being plasmolysed when they are placed in what? Hypertonic solution. Exosmosis occur. Exosmosis in the sense that water moves from inside of the cell outside the cell. That's the idea. Hemolysis is the reverse. You are placing the cell inside hypotonic solution whereby water is more outside than inside. So water will move inside the cell. Then if water move inside the cell gets bigger, becomes turgid, then burst. That's hemolysis. And a very, very good example of organism that does this is what? Is your red blood cell. Let's note that. Then the last one now is no, not the last one. Turgidity. Turgidity is just before hemolysis can occur, before the breakage can occur, water will first get into the cell, right? So the after effect of water getting into the cell is that the cell becomes more turgid, more swollen before it bursts. At that point, you say the word the cell is turgid. That's turgidity, basically. Then flaccidity is when it shrinks before kind of breaking. That's the idea. So another thing I want you guys to also note is this: for plasmolysis, for both plasmolysis and hemolysis, the word lysis means breaking down. So for plasmolysis, it shrinks and then started what? Breaking down. Then for hemolysis, it's burst. Of course, that's automatic breakage. And that's that about cell and its environment. So thank you.